Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Genomics and Machine Learning for the In Vitro Sensitization Testing of Challenging Chemicals. My name is Sonia Hunt, and it's my pleasure to be your X-Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Sensitization, who developed the content for this presentation. Sensitization is an innovative biotech company that develops and sells in vitro testing methods for safety assessment of various toxicological endpoints across different industries. The tests replace animal experiments with a genomic-based testing platform, combining pattern recognition and machine learning, providing high informational content and a holistic view of the immunological responses, resulting in high predictive accuracy while supporting product safety and cost efficiency. The GUARD test portfolio includes assays for skin and respiratory sensitization, helping companies through the hurdles in product lifecycle. Hundreds of samples, including difficult-to-test samples, have been tested with GUARD TM and successfully reported. Sales are performed by partners, Sensitization's headquarters in Sweden and its sales office in the USA. Now, it's my pleasure to introduce your speakers for today's event, and I'd like to introduce you to Dr. Andy Forride, Scientific Liaison at Sensitization AB. Dr. Forride holds a Master of Science in Molecular Biotechnology from the Faculty of Engineering at LTH Lund University and a Doctor of Philosophy in Immunotechnology from Lund University. He has many years of experience working in vitro assay development, genomics, and machine learning. He is also a co-developer of the GUARD platform for the assessment of chemical sanitizers and is the inventor of 2 patents related to the technology. Our second speaker is Dr. Henrik Johannesson, and he's a chief scientist at Sensitization AB. Dr. Johansson is a life science professional with more than 10 years of research and development experience in the fields of immunology, toxicology, and cell and molecular biology. Specialized in vitro assay development for predictive toxicology and a co-developer of the GUARD platform, Dr. Johansson is passionate about bringing the latest advances in technology to practical use for the benefit of society. Now, before I pass over the controls to our speakers, I would like to first launch our first poll question, and you should see it appear in front of you momentarily. And there it is. Now, this is done in real time, so your participation is greatly appreciated, as I will be able to share the results with you once I close the polls. So if you can go ahead and cast your votes right now, you could pick on any of the below, select all that apply, and the question is, what are your testing needs, or what are your testing needs? R&D, e.g. example, internal data for decision-making, regulatory compliance, marketing claims, occupational health, or other. So please go ahead and cast your vote, and then I will close the polls in about 10 seconds and share the results with everyone, and then I will pass it over to our speakers to begin their presentation. So once again, that question is, what are your testing needs? R&D, internal data for decision-making, regulatory compliance, marketing claims, occupational health, or other. It looks like we have the majority of our audience members have voted. So thank you very much for that. And for those of you who have not, please do so right now. In 5 seconds, I'm going to close the polls. Okay, closing the polls. And now I'm going to share the results. And the results of our first poll is we have 55% at R&D, internal data for decision-making, 58% at regulatory compliance, 5% at marketing claims, 18% at occupational health, and 23% at other. So thank you very much, everyone, for participating in that. Now I'm going to pass over the controls to our speakers. You may begin when you're ready, doctors.
Thank you, Sonia, and thank you all for joining. It's great to have so many attendees at this webinar. My name is Henrik Johansson. I'm a chief scientist at Sensitization, and I'll actually start with giving a brief introduction to our company. It was founded in 2010 as a spin-out company from Lund University, and we still have our headquarters in Lund, Sweden, with additional sales offices in US and France. Sensitation's main line of products are the GARD assays for in vitro assessment of various toxicological endpoints, primarily different types of sensitization. We do continuous product development, aiming to both sharpen and broaden the applicability of our assays, which we also offer as a service for sponsored studies. The GARD assays are also available through contract laboratories and distributors worldwide. The GARD assays, which will be the main focus of this presentation, are broadly applicable in chemical, cosmetics, pharma, and medical device industries, and have repeatedly demonstrated very high predictive performance. And our long-term vision is to be able to establish a new industry standard. So the prevalence of allergies is continuously increasing. Skin allergies are the most common type of immunotoxicity in response to chemical exposure. Today, more than 20% of the population are affected by allergic contact dermatitis in developed countries. And while respiratory sensitization is not as common, the implications are severe with potentially dangerous outcomes. So for these reasons, assessment of chemicals in terms of their ability to induce sensitization is a crucial part of safety assessment. Historically, such testing has been made primarily using animal-based methods. The murine local lymph assay is still the preferred method in certain industries and regions, but today we see a shift towards more and more non-animal-based and alternative methods. This shift is driven by a number of diverse factors, including regulatory, ethical, scientific, and economic factors. Research in this area was definitely accelerated by the ban of animal-based methods in the cosmetics industry in the EU, a trend that is now spreading both globally and across market segments. But the trend is also driven by increased community engagement and cost and effectiveness considerations. So today there are a number of validated in vitro methods available, but there are a number of considerations to make when doing this kind of testing, and the decision-making is rarely straightforward. We know that available methods are not recommended for standalone use, partly due to insufficient predictive capacity and possibly inconclusive results. This in turn may lead to increased costs associated with integrated testing strategies and defined approaches. Another concern is the applicability domains of each method and their usefulness for difficult to test substances. There's also a lack of potency classifications in vitro, and for both in vivo and in vitro testing, there is a lack of methods for specific assessment of chemicals inducing sensitization of the respiratory tract. So a great deal of progress have been made in a relatively short period of time, but there is definitely room for innovation within this field in which Sensitization is placing its core business. So against this background, I would like to introduce the GARD technology platform. GARD stands for Genomic Allergen Rapid Detection, and we view GARD just as a platform which combines the human immune system with high informational content genomic readouts and machine learning assisted classifications. Based on this platform, we today have a number of different individual assays, and they include GARD Skin, which is a hazard identification method for skin sensitizers, with the add-on of GARD Potency, which is a sub-classification method for identified sensitizers according to the relative sensitizing potency, weak or strong. GARD Skin medical device is an adaptation of the GARD Skin method suited for testing of solid materials, including medical device. And then we have GARD Air, which is a novel assay for assessment of respiratory sensitizers. I will come back to each of these assays later on in the presentation, but I will start by introducing the concept of the platform. So we view all of the assays as separate entities, but they are all based on the same technology. And conceptually, the GARD platform can be viewed as a modular testing strategy framework based on interchangeable modular components. These components are the biological cell system and a training data set used both for biomarker discovery and subsequent training and definition of the prediction model. And then you have the genomic biomarker signature, which is specific for each assay and related to the mechanistic events associated with the individual toxicological endpoint being investigated. And then lastly, you have the prediction model, which performs the final classification and interpretation of the high dimensional data generated in the assay. So each of these modules can be independently interchanged and designed in order to customize assays for specific toxicological endpoints. So before returning to the individual GARD assays, I will go through each of these components from a method development point of view in order to illustrate both how each method was developed and how they work once they are finalized. So if we start with the biological cell system, it's of course helpful to know at least something about the mechanistic background of the toxicological endpoint you're investigating. And to this end, the AOP, or the adverse outcome pathway, may be of some assistance, illustrated in this figure. So this is quite common knowledge today, and much have been learned about the process of sensitization within the last 10 to 20 years. But in a brief summary, sensitizing chemicals penetrate skin and form protein-hapten complexes due to their reactive profiles in contact with proteins. These hapten proteins are taken up by dendritic cells, which mature and migrate to the local lymph node, where antigen presentation occurs to primarily specific T cells. So once primed T cells are available in a sensitized individual, upon secondary exposure, you will have a very rapid elicitation of the allergic responses. So the GARD platform utilizes the decision-making and the attenuation and initiation of downstream responses of the dendritic cells. And if we look at the dendritic cells, we today know that these cells react in a multitude of different ways in contact with these types of chemicals. You have recognition of foreign substances through innate immune receptors. You may have activation of self-defense mechanisms, such as upregulation of co-stimulatory molecules, including CD86. You may have cellular stress responses, as well as metabolic and oxidative stress responses, and of course, communication with different parts of the cell system, including chemotaxic receptors and antigen presentation and functionalities of these cells. And all of these functions are, of course, controlled by the proteome of the cells, which is in turn regulated by the transcription from DNA to RNA. So this is the basis and one of the founding reasons that why we believe that transcriptomics is a very efficient way to get a holistic view of everything that's happening within these cells in response to chemical exposure. So with this knowledge in mind, the underlying hypothesis for each GARD assay is basically that there should exist genes and pathways in dendritic cells that are differentially expressed depending on the stimuli, whether or not it's a sensitizer or a non-sensitizer. And if we can find those genes, they can be used as predictive tools. So in order to test this hypothesis, each method development also starts with the definition of a training data set, or the creation of a training data set. And basically what we need to do is to identify a training reference set of chemicals with known properties relating to the investigated toxicological endpoint. So as an example, you can see on the right here an outtake of the training data set for GARD-SKIN method. And having access to this training reference set of chemicals, we can create whole genome data, which can be mined for further information relating to the endpoint being investigated. Which leads me to the identification of a genomic biomarker signature. So having access to whole genome data generated from this reference set of chemicals, we can use statistical and computational methods to mine this data set in order to identify the genes that are differentially regulated in the two different groups of reference chemicals. And having access to a specific genomic biomarker signature, we are also able to train a computational prediction model, primarily using support vector machines that can assist in the interpretation of the high informational and high dimensional data that is being generated within each GARD run. So having all of these different modular components in place, we can define and freeze a finalized assay. And once we get to that stage, every GARD assay more or less follow the same protocol, which can be summarized in the following 6 steps. So firstly, we identify a suitable concentration in which we can expose the cell cultures of the test item. We do so by monitoring the cytotoxic effects induced by the chemical. Once we know what concentration to use, we can repeat exposure experiments in 3 biological replicates, and from these samples we can extract the genetic material or the RNA from these samples. Having access to the RNA, we can then perform gene expression profiling, and then we can actually quantify the expression levels of each of the biomarkers of interest for each individual GARD assay. Having access to the high dimensional data, we can feed this into our web-based applications, which holds the prediction models and the computational algorithm with output a classification stating whether or not it's a sensitizer or a non-sensitizer. And again, we have these 4 different assays readily available for use today, and I'll go through each of them more in detail and just talk briefly about the performance of each of these assays and where they are in a regulatory acceptance point of view. So starting with GARD-SKIN and GARD potency, as I mentioned in the beginning of the presentation, GARD-SKIN is a binary hazard identification for assay for skin sensitization. It's based on 200 genomic biomarkers, and it's been shown to be very accurate and sensitive and specific. While it provides binary hazard identification, if we combine it with GARD potency, we can actually achieve a full risk assessment of tested items, as the GARD potency assay provides subclassification of identified sensitizers. If we look at the data that are currently being in review by the ECVAM Scientific Advisory Committee, we can see that in a ring trial comprising Sensagen Core Lab and 2 naive laboratories, and using a data set of close to 30 compounds, the GARD-SKIN assay is very accurate with a 94% accuracy. It's also reproducible between labs and within labs, as can be shown in this figure. And what we see here is actually the majority vote from 3 individual experiments, but I'd like to also show you the raw data signals, which is the support vector machine decision value output from each experiment. And I do this to show you that it's also reproducible at signal level, both across the 3 different laboratories involved in this ring trial, but also across 3 individual repeated experiments that were made within the ring trial. So combining this with guard potency in what we refer to as a guard tiered approach, we can also then subclassify identified sensitizers according to their potency. And we did this in this same data set used for validation. So these are the expected sensitizers used within this ring trial. And you can see here the performance of guard potency in subclassifying these chemicals according to their relative sensitizing potency. And we could conclude that guard potency is 88% accurate based on this data set. So combining these 2 methods into the proposed guard tiered approach, we can actually provide full risk assessment according to GHS and CLP with roughly 80 to 90% accuracy. And all of this data has very recently been accepted for publication in toxicological sciences, and hopefully it's currently in print and will hopefully be out in just a couple of weeks or so. So moving on to the medical device protocols, these are actually add-on protocols to the existing guard skin methodology. So this is an adaptation of the protocols enabling us to test solid materials, and we do so in accordance to the ISO 10993, which states that both polar and non-polar extraction vehicles should be used in order to extract leakables from solid material. And we have tested this by spiking material using both silicone and TPU with known sensitizers and non-sensitizers, and extract these leachables from these materials using the recommended protocols from ISO 10993. And we were able to accurately classify all of the spiked in materials. So this is showing great potential to be used in medical device industry and also other industries concerning solid materials. And then finally we have guard air, which is a novel and experimental assay for assessment of respiratory sensitizing chemicals. It's based on 28 genomic biomarkers, and we propose that we can use this standalone or in combination with guard skin to discriminate between respiratory and skin sensitizers, a feature that has previously not been possible using animal-based methods. So also guard air has been the subject of a ring trial using the same laboratories. And you can see here that among the expected positives, we have a few false negatives, but on the other hand, the method is very, very specific, meaning that we have extremely low number of false positives. So currently we propose that this method can be used for opt-out applications within R&D and decision-making at an early stage. So to close this off, I'd just like to summarize the current status in terms of compliance and regulatory acceptance. So guard skin and guard potency are currently being reviewed for validation. It's included in the OECD test guideline program. And guard data is also accepted by ECHA for REACH dossiers. Guard is also being audited and has been audited quite recently for GLP compliance. And this was finalized during this spring, and we expect an approval within this next couple of weeks, actually. So those are the introductory slides to the guard platform as such, and I will leave the word over to Andy to talk about how these assays can be used in industry. But first, I think I'd like to hand over to you, Sonia.
Yes, Dr. Johansson, yes, it's time now for our second poll question for our audience members to engage with. You'll see it right there in front of your screen. I'll give you about 35 seconds to complete this poll question before we hand it over to Dr. Johansson. So the question is, what are your top 3 challenges related to sensitization testing? Now, please select one of the below. The options are difficult to test samples, lack of in vitro methods for CLP classification, performance issues, for example, accuracy. The next option is ethical issues with animal testing. And then the last option is other. So please go ahead and cast your votes. As you know, this is done in real time, and I'll be able to share the results with you once I close the polls. So your participation is greatly appreciated. And it looks like we have the majority of our audience members have cast their votes. So thank you very much for that. For those of you who have not, please go ahead and do so. I'll give you another 10 seconds, and then I will close the polls and share the results. Okay, thank you very much, everyone. Looks like we got a lot more people jumping in now to cast their votes, so I do appreciate that. Okay, I'm now going to close the polls and share the results with everyone. And here's the results. So we have 27% said difficult to test samples, 18% at lack of in vitro methods for CLP classification, 27% said performance issues, for example, accuracy. We have 20% at ethical issues with animal testing, and 7% for other. So thank you everyone for that. And now I'll pass it over to Dr. Johansson.
Thank you, Sonia, very much. So this is actually not Dr. Johansson, this is Andy Forryd.
Oh, Dr. Forryd, yes. Well, there you go, Dr. Forryd. You may go ahead.
Thank you very much. So I'm taking over here from Henrik. So thank you, Henrik, very much for introducing us to the GARD platform and also to the different assays. So when I now move on to the next part in this presentation, I would like to focus less on the technical aspect of the assay and instead try to describe how our customer has used the GARD assay in different type of applications. So I will present a selection of applications and case studies where we describe the tailor-made adaptations of the assay protocols in order to adapt to the customer's requirements. And I will also demonstrate the usefulness of these applications in real-life customer project. So in the first part, I will present cases related to the testing of samples that has traditionally been considered as difficult to test, or even outside of the applicability domain of the current in vitro assays. So there's of course many different factors that can make a substance difficult to test. It can, for example, be pre and pro-haptens that need to be activated in order to acquire skin sensitization potential. And the test system may not actually have this required metabolic activity. It may also be related to solubility issues of the specific substance. That may limit the test concentration you can use, and you can also then risk to have false negative classifications. So you may also have different type of surfactants or cell membrane damaging substances that may be very cytotoxic to the cells and risk to give rise to false positive predictions. And finally, I mean, we should not forget that the available in vitro assays primarily defined for the testing of neat chemicals, and their applicability to the testing of complex mixtures and formulation is not well described today. So the goal with the case studies of part 1 is essentially to evaluate the applicability of the GARD assay for testing this type of challenging substances. And we would also like to describe how we have extended the GARD protocols in order to enable for routine testing of samples that are not soluble in the conventional in vitro assay vehicles, which is then DMSO and water. And finally, we would also like to show you a case study from a client where we actually used the assay to test a subset of very hydrophobic substances that have been considered outside of the applicability domain of current in vitro assays. So in the first case study, the aim was essentially to evaluate the applicability of the GARD skin assay to test pre and pro-haptens. and also substances with low solubility in water. So what we essentially did was we identify a subset of pre and pro-haptens and substances with low water solubility, which is here defined by having a high log p value. And we also made sure to see that all of these chemical had good reference data from the OECD validated assays, and also that we have tested them in the GARD assay. So as you can see from this slide, the majority of these compounds were actually quite difficult to test in the conventional in vitro assay. When we evaluate the performance of the GARD on this subset of chemicals, we could see that we had a 92% accuracy and only misclassified two of the chemicals. So the overall conclusion from this comparison was that the dendritic cell-based system, meaning both the H-CLAT but also the GARD assay, was well applicable for the pro and pre-haptens. And we could also, when we look at the gene expression data from the sensor cells in the GARD assay, we could actually confirm that they are actually expressing the metabolizing enzymes that may be required in order to convert the pro-haptens. So concerning the samples with low solubility, these were also quite well predicted in the GARD assay. And we think that one of the explanation could be that the output that we measure in the assay, mainly the gene expression profile, is very sensitive, meaning that we only need to have a very low concentration of the test item in solution in order to be able to provide a reliable result. So in the next case study, the aim was essentially to verify the observation from the first case study on a new test data set. And we would also like to compare the results from the GARD assay to the reconstructed human epidermis assays, because they have also been suggested as one of the solutions to the solubility issues. So for this purpose, we essentially tested all of the 12 chemicals from a recent publication by Melling and colleagues, which investigated the applicability of reconstructed epidermis assays. And we retested all of these substances in the GARD assay. So these substances were a mixture of pre and pro-haptens, substances with low water solubility, and also a few surfactants. And as you can see from this very limited data set, the GARD assay performed quite well. We observed an accuracy of 80%, and this was better than the conventional in vitro assays. And it did also outperform all of the reconstructed human epidermis assays as well. So one of the incorrect classifications in this data set was a surfactant, the tween 80. So since there is very few surfactant present in this data set, we also decided to expand the number of surfactants that we have tested in order to make sure that we can also test this type of chemicals. So in addition to the tween 80, we tested an additional 8 substances that were surfactants, and we could see that the assay could actually perform quite well also for this type of substances. So in order to conclude the case study 2, the GARD assay had a predictive performance of approximately 80% when we compare to existing human data, and that outperforms the reconstructed human epidermis assays as well as the conventional in vitro assays. We also again observed that pre and pro-haptens and samples with low solubility actually performs very well in the assay. And we could also see that 89% of the surfactants were actually accurately predicted. And that also include substances that is such as SDS and benzalkonium chloride that has traditionally been considered as very difficult to test in vitro system. So the aim of the third case study was essentially to further define a protocol on how we could actually test substances that are completely insoluble in water and DMSO. So the main idea with this was essentially to evaluate a selection of solvents that were less polar than the conventional in vitro assays vehicles that we use. And we do that in order to be able to actually select an appropriate solvent. So as you can see here in the table, that's basically summarized the different solvents that we evaluated in this study. And as you can see, these solvents could be used at a concentration between 0.25 and 1%. And when we actually test the pure solvent in the GARD assay, we can see that they were all predicted as non-sensitizers. But when we spiked these different vehicles with a skin sensitizer, we were able to also pick up the sensitizing signal in the different vehicles. And as you can also see from the heat map, the gene expression profile of the non-spike samples and the spike samples give rise to a very similar gene expression profile, which indicate that they did not interfere with the assay result in the end. So this slide essentially demonstrates the importance for the test results to select an appropriate solvent that is based on the physiochemical properties of the test substance, and also how this strategy with alternative vehicles can actually increase the solubility and bioavailability of the test item in order to prevent false negative classification. So these figures is from a real client case where the customer actually provided us with a substance that were not soluble in DMSO or water. So in the two figures up to the left, we try to get this compound into solution into either DMSO or water, but the compound was completely insoluble using these vehicles, and the substance were also later predicted in the GARD assay as false negative. However, we considered the physiochemical properties of this test item. We decided to use a combination of less polar solvents. In this case, we used DMF and glycerol. And as you can see, we could observe a drop in the relative cytotoxicity of the cells, and we could also finally get the compound into full solution, and we could predict the substance correctly as a skin sensitizer. So this kind of highlights the strategy we use in order to test these substances with very low solubility in the conventional vehicles. So in the final step of case study 3, we applied this extended protocol in order to evaluate a subset of 7 UVCB samples that were extremely hydrophobic and not applicable for testing in the other conventional in vitro assays due to solubility issues. And the reason why the client would like to test them is because they already had animal data available, but now they would like to further explore if they could use in vitro assay testing instead. So when we tested these 7 UVCB samples, we could see that all of the 7 could go into solution. We were able to test them at an appropriate concentration, and we could correctly predict 6 of those 7 substances in the GARD assay. And most importantly, this was quite conclusive results, and we did not report any false negative due to solubility issues. So in the final case study for testing of the difficult to test substances, I would like to also describe how we do when we test complex mixtures and finished formulations. So in this specific case, we had a customer who requested the testing of e-liquids. So e-liquids is quite complex to test in vitro. They comprise of a base formulation that often contain a flavoring ingredients and nicotine. And nicotine can be quite toxic to the cells. So from a toxicological perspective, the main ingredient of concern is the flavoring ingredients, because that can sometimes show sensitizing potential. So the problem is basically to be able to pick out this signal among the other components present in the mixture. So the customer provided us with 3 base liquids that contain different type of different concentration of nicotine, and they also provided us with 2 commercially available e-liquids containing both nicotine but also flavoring ingredients. So we tested these in the GARD air assay for respiratory sensitization, and they all came out as non-respiratory sensitizers. We tested the same compounds also in the GARD skin assay, and only the 2 compounds containing in the flavoring ingredients were classified as skin sensitizer. And these were also confirmed in the GARD potency assay as weak sensitizers, class 1B, according to the CLP regulation. So this study has also now been published in a Journal of Regulatory Toxicology and Pharmacology, and I think this is quite a good strategy on how to actually test different type of formulations. So this essentially concludes the first part of difficult to test substances. And just to basically summarize, I think that, I mean, the GARD assay is applicable for a wide range of different type of chemicals and applications. And we also now demonstrated that it works quite well for different type of challenging substances. So today we have tested approximately 400 substances in-house, and the reported accuracy is similar across the industrial application and also across the different chemical reactivity domains. But that being said, I mean, there's still new areas to explore, and we haven't generated a lot of data on all of this group. And we are, of course, always open for different type of research proposals and collaborations in order to further explore the applicability domain of the assay. So by that, I would like to move on from the first part and switch focus from testing of difficult to test substances, and instead describe how the GARD assay can provide valuable information throughout the different step in the product life cycle by presenting different type of case studies from each of the different phases, all the way from product development to regulatory fillings, production phase, and all the way to marketing in order to support the marketing claim. So I would like to start by describing how the GARD assay has been used during product development step, and to describe also those tailor-made adaptations that we have done to the protocols in order to live up to the requirements from the client. So in this specific case that I would like to present now, we had a customer from the chemical industry, and they have generated a lot of animal data, but they would like to transfer from animal results to in vitro testing due to their corporate social responsibility policy. So the customer is really used to look at the local lymph node assay data, but the aim of their study was essentially, or their requirement is essentially, to have an assay that are capable of rank candidates based on their skin sensitizing potency during their R&D phase, in order to identify and remove the potentially problematic compounds and to prioritize compounds for further use. So this is something that is not currently applicable for testing in the conventional in vitro assays. So in order to meet the client requirement, we've actually modified the GARD skin protocols. So instead of performing the testing only at one fixed concentration, we perform the GARD skin assay at different concentrations in order to generate a dose response curve. So we were interested in actually finding the concentration, or the lowest possible concentration, giving rise to a positive classification in the assay. So this is what we refer to the decision value 0, or DV0 value, and that is analogous to the local lymph node assay EC3 value. So if a low dose is required in order to induce this value, that means that the compound has a very high potency, and also vice versa. If you require a high dose, the compound has a low inherent sensitizing potency. So in a pre-study that the client performed, they tested a total of 4 substances where they already have animal data available in order to compare the results. So what we could see is that all of the test items demonstrated a very clear dose response behavior in the GARD dose response model, except of course for the non-sensitizers, which did never pass the threshold. And these figures are also sorted based on the relative sensitizing potency from the non-sensitizers all the way to the bottom right, where we have the strongest sensitizer. And what we can see is that the stronger sensitizers typically induce a DV0 value at much lower concentration. And if you compare this to the ranking from the local lymph node assay, the ranking provided by the decision values is similar to the data provided by the local lymph node assay. So this was very useful for the client. They could use this data in order to rank their compounds based on relative sensitizing potency, and that helped them to facilitate their internal decision making and in order to prioritize the compounds for further research. So this initial testing, actually, when we saw this data, we were really interested also to see how well this strategy would work on a larger data set. So what we did was essentially to use the same strategy. We evaluated a larger data set of 30 compounds, and we correlated the data to the local lymph node assay EC3 values, but also to the human potency categories reported by David Basketer. And we could see that we could actually observe significant correlations between the decision values generated in the GARD assay to both local lymph node assay EC3 values and human potency as defined by David Basketer. So one tentative use of this approach is, of course, that you can perform your testing in the GARD assay. You can identify the decision value. So in this example, say for example, we have a DV0 of 10 micromolar, and then you can use this information in order to extrapolate a local lymph node EC3 value. In this case, it's classified as a moderate sensitizer with an EC3 value of 1.5%. But you can also extrapolate the data to actually predict the human potency class, which in this case was class 2. You can also, of course, use the EC3 values as a point of departure for defining your risk assessment procedure. So this is some extremely novel data we have generated, and we will present this data in a webinar on June 17. So I hope you will be able to participate in this webinar. So please contact me, and I will be happy to send you an invitation also to this webinar. So in the next step, I would also like to show you how the GARD assay can be used for regulatory purposes. So in this specific project, we were approached by a client that had to complete the REACH registration dossier with skin sensitization data, and they had a compound that were not applicable for testing in the OECD validated assays due to solubility issues. So as my colleague Henrik already mentioned, the GARD skin and potency assays included in the OECD testing guideline program, and the results can be used for REACH registration in accordance with the Annex 11 of the REACH registration. So this testing was performed at our CRO partner Eurofins in Germany under GLP condition, and we could get the sample into solution into an acceptable concentration. The compound was classified as a non-sensitizers. The results were submitted to ECHA, and the study was accepted without any restrictions as well. So in the next case, I would like to show you how the assay could also be used during production and to check different batches during the production of samples. So in this specific case, we had a client within the medical device industry. They had produced a new batch of their products, and once this batch actually came out to the market, they had several complaints on allergic contact dermatitis from clients. So they suspected that there was something wrong with this batch. So they asked us to test both the old batch and the new batch in the GARD skin medical device assay. And in this testing, we could actually confirm the data they had from the clinical experience of the individuals, because the new batch were classified as a sensitizer, while the old batch was still classified as a non-sensitizer. And this was also later traced by the client to impurities in the new batch of the products that might be introduced during a change in the sterilization process. So I think this case basically highlights how you can actually use the GARD assay also for proactive testing when you do some modifications in your production phase. So in the final step, in the final case I would like to present today, I would like to close this circle because I would also like to present a case where client has actually used the data generated on the GARD assay for products that have already been marketed in order to support the marketing claims. So in this specific case, the client would like to test some of the finished cleaning products. So in this case, there were 3 commercially available cleaning products, and they were requested by local authority to provide scientific evidence in order to support their marketing claims. And they were also asked to complete their safety data sheet because these products contain skin sensitizers, and now they also requested the information regarding respiratory sensitization. So we performed this study in both the GARD skin and the GARD air assay, and as you can see, 2 of the compounds were classified as skin sensitizers in the GARD skin assay, which was also consistent with what's already were included in the SDS sheets. But all of the compounds were classified as non-respiratory sensitizer in the GARD air assay. So the customer used the final report as evidence to strengthen the product claim, which was also accepted by the local authorities. So by that last case, let's basically conclude this presentation. And of course, if you're interested to hear more, you're always welcome to contact us, either us at Sensitization directly or any of our CRO partners around the world. So in addition to the Sensitization lab, we do also have collaboration with Eurofins in Germany, Burleson Research Technologies, and MB Research in the US. They are fully equipped GARD laboratories, and they are capable of performing the full study for you. In addition, we do also have a broad network of different distributors that will, of course, be able to assist you with your testing and to be able to answer your questions. Of course, also, please do not hesitate to contact either myself or my colleague Henrik if you have any questions relating to this presentation. And you're, of course, also welcome to visit our homepage, which contains recorded versions of previous webinars, and they also contain a lot of information regarding the services that we provide. So by that, I would like to thank you very much for listening, and Henrik and myself will be very happy to take any questions. So I would like to leave it over to you there, Sonia.
Okay, thank you very much for that great presentation. We have our final poll question right now to launch, so we really would encourage our audience members to participate in this last poll question, and then we'll jump right into the Q&A portion. So here's a question. In today's webinar, which were the top 3 interesting topics for you? So please select just one, the GARD technology and how it works, features and benefits of the GARD assays, case study 1, GARD assays for difficult to test samples, case study 2, GARD assays for product life cycle, or case study, GARD dose response. So please go ahead and cast your votes. It looks like we have the majority of our members have done so, so I do appreciate that. And for those of you who have not, can you please go ahead and do so? I will give you another 10 seconds, and then we're going to jump right into the Q&A. Okay, so thank you everyone for participating in that. I am now going to close the polls and share the results. Okay, so here's the results. 18% the GARD technology and how it works, 24% at features and benefits of the GARD assays, 26% at the case study 1, 5% at the case study 2, and 26% at the case study GARD dose response. So thank you very much everyone for participating in that. And now I'm going to start the Q&A portion of the webinar. So if you haven't done so yet, please go ahead and do so right now. You can go ahead and pass your questions to me using that questions box, and we'll try to attend to your questions during the Q&A session. And so now I have a couple of questions already lined up. So Dr. Fouweride and Dr. Johansson, are you ready?
Yes, Sonia, please go ahead.
Okay, so the first question I have is, how do you foresee the dose response method to be used moving forward?
Yeah, so I think that, thank you very much for that question. That's very interesting. And I think, I mean, this is something that we will present in the webinar we will host at June 17 as well. So I think this is a really interesting strategy as well, because I think this is the first time that you can actually use in vitro data in order to actually derive a point of departure value for risk assessment. And I think, I mean, those that are interested in this should definitely join our webinar on June 17, because I think this is a really interesting strategy. And you can actually use this value both to extrapolate to human potency, but also take the route through the local lymph node assay value, dependent on your requirements.
Okay, thank you very much for that. I'll go into our next question here. I got a couple of them lined up, so let's see if we can get through them. Is there any mechanistic explanation to the observed high accuracy of GARD?
Hi, Sonia. Thanks. I think that we can identify a number of technical aspects in the GARD assays that are unique to our strategy of testing, and that would be firstly the high informational content in using transcriptomic analysis, enabling us to get a holistic view of a lot of mechanistic events in response to chemical exposure. And then secondly, I would say that we identify the biomarker specific for each toxicological endpoint using a data-driven approach. And I mean that as opposed to a hypothesis-driven approach. So we can actually learn from the generated data in the training data set what measurable effects we can identify and base our developed assay on that information. And lastly, also, I still believe that, I mean, interpreting this high-dimensional data using a machine learning-based classification system is still also quite unique for GARD. So I think those are the main points that I believe would explain the observed high accuracy that we have obtained for these assays.
Okay, thank you very much. Here's the next question for you. Do you have any idea if the HY ethylene diamine was negative in GARD skin by the 3 test labs, whereas if it had been classified as a skin and respiratory sensitizer?
Yeah, I mean, we suspect that this might be, I mean, this is a very special chemical, and I know that several of the other assays also have problem with just ethylene diamine. I think this is a slow-acting prohapten, so it's there might be something related to the kinetics of the reaction as well. So maybe we need to, in order to actually capture this, we might need to increase the incubation time. But that's, I mean, that's something we haven't investigated further. But that might be due to the slow kinetics of this substance as well.
Okay, I'm going to try to squeeze in a couple more questions. We got a lot of questions coming through here, so let's try a couple more here. Here's the next one. Have you tested the GARD system with combinations of chemical sensitizers? Is your platform able to predict combinatorial effects?
So I don't know if I have a good response. No, that's fine.
Okay.
Thank you, Sonia. I'm not sure if I have a good response, though. I think that provided that the concentration are sufficient in the mixture or the product that you're testing, I do not see any problem with that. But I think that the real issue there is really associated with the mixtured nature of the test item, which could potentially provide some problems when defining what concentrations to use for testing. I'm not sure if that answered the question, but please come back to us and we can discuss that more, whoever asked the question, and we'll try to explain this better.
Okay, thank you. Here's the next question I have for you. Did you recently test more typical skin sensitizers in GARD air and vice versa, typical respiratory sensitizers in GARD skin?
Yes, thank you for that question. So the validation ring trial study for GARD air actually included quite a few skin sensitizers. And they were all classified by GARD air as non-respiratory sensitizers, which we believe are very interesting data and correlate quite well with the clinical experience of these chemicals, as they are pretty much always either a skin sensitizer or a respiratory sensitizer. I mean, the double types of sensitizers do exist, but they are quite uncommon. So this really proves that GARD Air is specific for respiratory sensitization, and it does not provide positive signals induced by skin sensitizers. We have also done vice versa. We have tested respiratory sensitizers in GARD skin, and we see the same patterns. It's quite specific. Again, we do have some false positives induced, but majority of the compounds are predicted as either or, which is also what's expected based on clinical experience.
Okay, thank you very much for that. We have a couple more questions here. I'll try to see if we can squeeze in these ones here. The next one is, do you have any experience with natural extracts?
Yes, we do. We have run a couple of natural extracts as sponsored studies. So that's something that we do have experience of, yes.
Okay. The next question I have, are these test methods accepted by any regulatory agency?
Yes, so in Europe they are accepted for REACH registrations according to Annex 11 in the REACH regulation. So that essentially means that you can use the test results for GARD skin and GARD potency as a weight of evidence approach for your REACH registrations. And that being said, I mean, we're also now being reviewed by ECVAM and then the scientific panel of ECVAM ESAC for full inclusion into the OECD test guidelines. So we expect this to open up some doors, not only in Europe but also in the US.
Okay, thank you very much for that. Here's the next question. Do you have any experience in plant protection products?
I guess that would depend on how you define plant protection products. We have experience with agrochemicals, meaning pesticides, but I'm not sure right now whether or not we have also tested plant protection chemicals, but that's something that we can definitely look into.
Okay, here's the next question. I'm going to just do two more questions here. What are differences between sensor cells and mute cell lines, MUTZ cell lines?
So sensor cells are derived from MUTZ3 cells, and for any listener who has been following the development of GARDs throughout the years would probably note that we have used both MUTZ3 and sensor cells to describe our cell lines in our scientific publications. We have always used the same cells, which originates from MUTZ3 cells, but we have cultivated them using in-house protocols, which is not based on the feeder cell line that is recommended for growth of MUTZ3 cells. So I would say that it's this prolonged use of an alternative cell media, which is only based on serum supplemented media, that has motivated this rebranding or renaming of the cells, so to speak. But it has always been the same cells that we have used and are still using today.
Okay, here's the next one. Are there statistics how good in vivo data can predict human sanitizers? Are these numbers better than the GARD results?
I'm sorry, could you repeat that? Sonia, I did not quite catch the question. Could you please repeat it?
Oh, sure, not a problem. Are there statistics how good in vivo data can predict human sanitizers? Are these numbers better than the GARD results?
Oh, absolutely. So there are a lot of reports from the last 20 years in which local lymph node assay primarily has been used as the gold standard for sensitization testing. And Most of these assessments conclude that LLNA is about 70 to 75% accurate compared to the clinical experience, meaning human data. And also the reproducibility is in the ranges of 70 to 80% according to these types of reviews. There are a number of good reviews quite recently describing and comparing in vivo and in vitro statistics relating to this matter. I'd be happy to share any recommended reviews if you get in touch afterwards.
Okay, all right, so this is going to be our final question, and here it is. How have you developed your data science approach? Is it from scratch using internal competencies, or do you rely on some ready off-the-shelf components to analyze your data?
I would say that we use in-house expertise and knowledge that we have acquired throughout the years, actually. Being based, I mean, Sensitization was a spin-out company from the university where a lot of competencies mixed and gathered. So we have acquired a lot of experience and knowledge within these research areas throughout the years. But I mean, they are, while not maybe off-the-shelf solutions, they are quite widely used in many different fields. Support vector machines are used pretty much in any data science application throughout society today, as well as the statistical approaches we use for the data-driven identification of biomarker discovery, which is basically based on high variate statistics and computational modeling, using a lot of open source programming packages that are publicly available and yeah, more or less off the shelf, as you put it.
Okay, all right, so this is going to be our last question. For regulatory purpose, example, REACH regulation, the GARD assay will have to be used in the AOP strategy combined with at least one other in vitro test. For difficult to test substances that cannot be tested with OECD 442, C, D, or E, guidelines that hasn't been developed yet for difficult substances, what do you preconize? What do you recommend there?
That is, of course, a very good question. I think, I mean, one thing to do is of course, to generate the data in GARD if it's possible, and then ask, I mean, you can complement that into a weight of evidence approach. For example, complement the data with the QSAR results or similar supporting information. And then, of course, you need to submit this data to ECHA because they are the ones who will decide if they accept the data or not. I'm not 100% sure how that would look from a regulatory perspective today, but I think that would be a valid approach to submit as much available data as possible, whether they come from testing strategies or from QSARs and supported by, for example, read across. I think that might be a valuable approach, but I think also that regulators need to be used to see this type of filings because, I mean, there is obviously a problem with the problem of testing difficult to test substances in the currently accepted OECD guidelines. Yeah.
Okay, well, thank you. Well, thank you very much for those questions, everyone. We have reached the end of the question and answer portion of the webinar. If we couldn't attend to your questions, the team at Sensitization may follow up with you after this presentation. If you have any further questions, please direct them to the email address that's on your screen. There you go, and you see it right there. Thank you, everyone, for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to this recorded archive of this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, I'm about to send you a link in your chat box. You'll be able to view the recording of this event at that link and also share this link with your colleagues when they register for the recording as well. So I encourage you to do that. Now, please join us in thanking our speakers, Dr. Andy Foririd, Scientific Liaison Manager, and Dr. Henry Henrik Johansson, Chief Scientist, both with Sensigen, for that very insightful presentation. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now.